Morning, everyone. It's nice to be here. First, are you seeing my presentation on screen? Are you seeing my?
Yes, yes.
Everybody
Yes.
seeing it?
Yes. Yes.
Okay, right. So, good
Okay.
morning, everyone, and it's nice to be part of this virtual conference. The title of my presentation is Plant Extract-Based Nanoparticles. A good perspective in nanomedicine. We all know that scientists have been using synthetic and physical means to get nanoparticles. However, recently, scientists have resorted to green chemistry in that they have used plant extract, which have been shown to be more environmentally friendly, bio-friendly, it's less expensive, and so forth. So this presentation will highlight to you the importance or the significance of green chemistry nanoparticles. Now, this is my abstract as was submitted to the conference team, conference organizing committee, and this is right. I shall begin. My introduction is, what is nanotechnology? We all know that nanotechnology is the design, synthesis, characterization of nanoparticles, which must have certain dimension. It must be between the 1 to 100 nanometer. If it's above that, it's no longer called a nanoparticles. And why is this very important? Within this dimension, matter show different properties in comparison to when it's of a large dimension. Right. Nanotechnology has found application in many realms. These include medicinal, medicinal field, biomedicinal field, such as antimicrobials, antibacterial, antifungal, antiviral. They have found application in cosmetic, food, feed, drug, gene delivery, environment, mechanics, optoelectronics, catalysis, light emitters, electrochemistry, etc. Research groups around the world have been working on, I've synthesized nanoparticles from metals. The ones that are commonly used are silver, gold, palladium, zinc, and that classification is increasing daily. Research groups around the world are intensifying their research in nanotechnology. So there is that spectrum of metal nanoparticles is increasing because of the importance of nanotechnology in the world of science. Now, out of these nanoparticles, the silver nanoparticles stand out. Why they stand out? Because they have unique properties such as chemical stability, good conductivity, catalytic, antibacterial, antiviral, antifungal, anti-inflammatory. They are found as component of composite fibers, superconducting materials, cosmetic products, optoelectronic devices. Silver nanoparticles have been used in wound dressings, etc. So silver nanoparticles have stand out because they are a versatile class of nanoparticles, performing roles in many realms. Now, before you can use your nanoparticle, you must design it and you must synthesize it. Synthesis can be divided into three categories. You can have physical synthesis, you can have chemical synthesis, and you can now have, in recent years, green chemistry synthesis. Now, the physical method include inert gas condensation, plastic deformation, high energy ball milling, ultrasound peeling. Now, the whole objective is to grind the particle to a nanometer size, below the 100 nanometer. So that will be from a top to bottom approach. Now, the disadvantage about physical synthesis is that particles are usually larger than 100 nanometers. So it's a cumbersome process. So it may not always give you that nanoparticle that you are looking for, the dimension and the morphology. In addition, physical approaches requires high temperature and high pressure, so it's very expensive endeavor. Now, the chemical synthesis of nanoparticles involve the reduction of chemical, phytochemicals, electrochemical, etc. Again, I mean, this is done worldwide, but it's an expensive method because it demands the purchase of reagents and so forth. The materials are usually toxic, so it's toxic to both the individual biologically, and it's also toxic environmentally. And in the synthesis of a nanoparticle chemically, you usually require a stabilizer or capping agent to prevent agglomeration of the metallic nanoparticles. Right? Because of the biological and environmental risks, scientists recently have resorted to use the green chemistry synthesis nanoparticles. And before I go on to the green chemistry nanoparticle, chemically, nanoparticle can be synthesized via the top-down approach or the bottom-up approach. The top-down approach starts from the larger size and grinding it down to the smaller nanometer size within the 100 to 100 meter nanometer, within the 1 to 100 nanometer range, using using acids, right? You can use acid to bring it down. That is the top-down approach. The bottom-up approach is that where you start with the atom or the finest particle that you have and you build it up to the nanometer dimension. So this is a synthetic route to the nanoparticles. You have the physical method, you have the chemical method, and you have the green synthesis. Now, the green chemistry nanoparticles have been found to be more safer, it's safer both to the person and to the environment. It's cheaper. In addition, you can scale up the synthesis as you would do for a chemical. And you know, it does not require the high temperature and pressure that you would do that are required for a physical synthesis. You can do nanoparticle synthesis at room temperature. Within 15 minutes, you can have your nanoparticles. In addition, the world has an abundance of forests. In my country, Guyana, I'm from South America, and our country has an abundance of forests. Green, it's very green, right? We are on the low carbon development strategy. We have been given the green light by Norway to keep our forests intact because of the abundance, so as to prevent global warming. So we have an abundance of forests in Guyana, and so using plant-based materials such as the fruits, the seed, the leaves, the root, whatever you name it, any part of the plant, even the peel, even a part of a plant that you want to throw away, you can use it and fabricate it into a nanoparticle. Basically, for a nanoparticle, what you need is, in the green chemistry synthesis, you need a plant extract, which functions as a reducing agent. You need a metal salt to provide that metal ion to become a nanoparticle, and you need a stabilizer or capping agent to prevent agglomeration of your nanoparticle. So, as I've said, almost all parts of the plants can be used. When you survey the literature at the current moment, there's over about 200 plants have been used, species. So research is intensifying in that direction, and it's good to see that scientists are moving towards green nanoparticle chemistry. Now, with respect to your metal salt, you can have different metal salt. If you want a silver nanoparticle from your plant, you can use silver nitrate. If you want gold, you can use gold chloride. If you want zinc oxide, zinc acetate, and whatever salt is easily to handle. If you need cadmium sulfide nanoparticle, you can use cadmium sulfate plus your plant extract. Zinc sulfide, you can use zinc sulfate plus your plant extract, and you just store it up at room temperature. And within 15 to 20 minutes, you can have your nanoparticles. You just filter it and do the characterization. Now, what in the plants are responsible for the production of nanoparticles? What in the plants? It's the natural products or the phytochemical constituents. They act as a reducing agent. They reduce the metal cation. here we see that we have a natural product, a phenolic natural product. Most of these natural products that function as nanoparticle precursors in plant are antioxidant in nature. They are phenolic, or they have hydroxy or phenolic groups, and so that they can donate, act as reducing agent. So in this case, we see this phenolic compound, two of it, complexing with Zn2+ ion, and then eventually, it releases a zinc oxide nanoparticle. So it's a very elegant synthesis. As I've said, there are over 200 plants of survey. When I checked the literature, Google search, over 200 plants have been worked on. For example, you look at this table. If you look at aloe vera, what plant part used as leaf? Nanoparticle synthesizes zinc oxide, so I presume they use zinc acetate. The size of the nanoparticle, 20 nanometers, so it's a nanoparticle. It's below the 100 nanometer range, and the morphology is spherical. Remember that in application of nanoparticles, you can have two nanoparticles of the same plant. One could be spherical, one could be in a different morphology, and they can have different medicinal application. Your application in medicine on the particle size and morphology will dictate what ailment they can prevent, because your nanoparticle morphology must be locked into the receptor sites. So there's morphology is very important. Nanoparticle morphology are very important in medicinal application and your dimension. So there is a whole range of plants that have been worked on: aloe vera, magnolia, cobos, the leaves, nanoparticle, copper. So I presume they use copper acetate, and a nanoparticle size 37 is spherical. If you took potato, they use zinc acetate. They got a nanoparticle size of 20. It's hexagonal. If you look at garlic, that we commonly use in cooking, you took the leaves of the garlic, they reacted with zinc acetate, and the nanoparticle size is 14, and it's spherical. And so it goes on, it goes on, like Cocos nucifera, they use the flower, they use silver acetate or silver nitrate, silver nanoparticles, 22 dimension. So you can use a wide range of plants or plant parts or plants that you throw away. Like if you're doing gardening and you cut your plants, you can take the plant parts and convert it into very useful nanoparticles. Now, as I've said, there are natural products or chemical constituents or phytochemicals in the plants that are responsible for the production of these nanoproducts. Now, it's beyond the scope of this select presentation to present to you all the structures of these natural products. These are some of them. Most are phenolic in nature, and they act as reducing agent. We see like galangin, epicatine. Look at these structures. Vanillic acid, they're all phenolic in nature. They have the oxygen donating electrons there. We have ferulic acid, eugenol, thymol. So they are more phenolic in nature or ketonic. They are antioxidant. They act as antioxidants in the plant. So they can be converted into they can promote the synthesis of metal nanoparticles. And this scheme shows a protocol for the synthesis of plant-based nanoparticles. Basically what you do, you take the plant part, you wash it, ensure that it has no microbes, you extract with water, you have your aqueous extract, then you add your metal salt. Within 50 minutes, you can see a color change. Then you do your filtration, and then you can do your stabilization or by capping to prevent agglomeration. And then after drying and so forth, you can do your characterization. Now in nanotechnology, there are several techniques that we can characterize the nanoparticles. We can use FTIR to tell you the different functional group, UV-Vis to show the absorbance of the nanoparticle, transmission, emission, microscopy, and SEM. We can use an X-ray diffraction to have the morphology of the nanoparticles. Right, so those are the techniques used. Now, once you made your nanoparticles, they can be used in medicine. As I've said, silver nanoparticles have strong antimicrobial, antifungal, and so forth. And what they have found is that in terms of antimicrobial activity, the nanoparticle, the surface area to the volume ratio is very important, because the larger the surface area, it's greater it's the ability of the nanoparticles to prevent microbial resistance. Right? Now, you may want to ask how these nanoparticles will destroy microbes. They act at the genetic level. They destroy the DNA and protein synthesis, you know, and so prevent replication of the microbes. And they have also found that silver nanoparticles is very useful in suppressing HIV virus because right, it's more effective in suppressing the HIV virus. They also have fungicidal effect because they can penetrate the fungal cell wall. Right? Plant latex cap silver nanoparticles have also been used in cancer. They have shown to inhibit human lung carcinoma cells. In addition, the nanoparticles can be transported to the cancer infected area. Right? Now, it is reported that gold and platinum-based nanoparticles may also have a significant effect on Wong Elin. Now, in Wong Elin, Wong Elin is said to be, you know, the mechanism is that you need to prevent. In Wong Elin, you need to promote tissue regeneration via an anti-inflammatory effect. Now, it is said that these plant-based nanoparticles promote Wong Elin via an anti-inflammatory effects. So they are used in Wong Elins. They have also found application as an antioxidant. You know, the antioxidants prevent the formation of free radicals, and we know that free radicals promote cancer, right? They induce abnormal mitotic divisions of cells. So your nanoparticles, which can act as antioxidant, can be used to prevent cancer via antioxidant by suppressing free radical formation. They have also measured the antioxidant capacity or evaluated the antioxidant capacity of certain nanoparticles, and they have found it to be greater than some conventional models, such as ascorbic acid. So these plant-based nanoparticles have very promising or have shown to be useful in nanomedicine. Nanoparticles have also been shown to have antiviral properties in that they inhibit the virus at certain stages in the life cycle. Some of them prevent the entry of the virus into the host cell. We all know that a virus cannot survive without a host cell. It needs a host cell to proliferate. And the nanoparticles can prevent entry of the virus into the host cell. And as I've said, nanoparticles, especially silver, have been shown to be effective against HIV pathogens. Plant-based nanoparticles have shown to have multiple binding sites to bind with GP120 proteins on viral membranes of HIV virus. This is what they have found. So it's very important in suppressing HIV. They have also been found to have protozoan, anti-protozoan effect. So they have been found to be useful in preventing malaria, right? This is what they have found. They have found that plant-based nanoparticles have been effective against malaria. Bearing in mind that they are the malaria plant, the plant that from which you have these anti-malaria drug, quinine, artemisinin, and aromatic, can also prevent malaria. The plant by itself can prevent malaria. That's a selective plant. The cinchonia bark. However, if you make certain nanoparticles, you can also prevent malaria by killing the protozoan involved in the life cycle there. Now, it's not only that, the medicinal realm is further expanded. Plant-based nanoparticles have been shown to have anti-diabetic activity in that they have a hypoglycemic effect. They can lower the blood sugar level. We know that there are two types of diabetes, type 1, type 2. The type 1 is dangerous where the pancreas cannot produce insulin. They have found that certain plant-based nanoparticles can prevent diabetes by lowering the blood sugar count. Right? It was shown that gold nanoparticles treated with diabetic mice show a decrease in the glycosylated hemoglobin level in normal range. So they have been found to have an hypoglycemic effect. Right? Further, in terms of application of nanoparticles in medicine, now nanoparticles can be also made from microbes. It can be made from bacteria, fungi, and virus, not only plants. And what they have found is that plant-based nanoparticles have been more superior than microbe-mediated synthesis. This is what they found. So this is another advantage of using a plant-based nanoparticles. So this could be very useful in a COVID infection. They have said microbes can act as a substrate. Maybe if we can get the virus to form nanoparticles, maybe we can in some way convert its deleterious effect into some useful effect. So who knows about this? Research needs to be continued in this direction. Now, if you survey the literature, there are many plants. As I have said, over 200 plants have been worked on to form plant-based nanoparticles. And these are some. I just give you a mini review of some. They have used Azadirachta indica. This is a neem plant. The neem plant is extensively worked on. So the neem plant has been a good source of plant-based nanoparticles and have shown good antimicrobial activity against gram-positive and gram-negative bacteria. Only 50 minutes was necessary for the synthesis, right? Gold nanoparticles have been, reacted with 4 different plant species. These are all listed here: Lipia, Citridoria, Pelargonium, etc., to form plant-based nanoparticles. And what is more interesting is that they can be used for imaging and drug delivery in the human biological system. So they're very important. Now, another application is that they have found that this leaf extract of Alicicarpus monilifer, after it forms a nanoparticle, maybe whatever, if it's gold or silver, whatever, I think it's silver nanoparticle, they use silver acetate, have been found to be effective against drug-resistant bacteria, such as methicillin-resistant Staphylococcus and coagulase-negative Staphylococcus aureus. Right? So silver nanoparticles from this plant, by taking this aqueous extract, reacting with silver acetate, filtering and and characterizing, have been found to be effective against methicillin-resistant Staphylococcus aureus and coagulase-negative Staphylococcus aureus. And this have been found to be very useful in preventing morbidity and mortality in HIV patients. In another literature search, zinc nanoparticles, zinc oxide, you can have zinc oxide nanoparticles were synthesized using a leaf extract of 2 medicinal plants, Cassia fistula, Melia, etc., using zinc acetate and the aqueous extract, 0.01 molar zinc acetate with the aqueous extract, and do your necessary reaction, filtering, and characterization. Now, what is the usefulness of this nanoparticles? Now, it is found to inhibit E. coli and S. aureus based on the disk diffusion assay. Now, if you are familiar with the disk diffusion assay, is an assay used to measure the efficacy of the antimicrobial activity of any antibiotics or any plant extract. Larger the zone of inhibition, greater is the antimicrobial activity. So they have found that the nanoparticles from these 2 plants have shown strong antimicrobial activity. Right? So that's a useful application. And in another application concerning silver nanoparticles, we have used the fresh leaves of the 2 plants, Balsamina and Lantana. And what they have found is that the nanoparticles from Lantana camara have shown antimicrobial activity comparable with standard positive controls such as ciprofloxacin, etc. So once it's comparable with the synthetic antibiotics, that's right away you can do an application there of the nanoparticles, providing it's safe. You know, that is what you want. When you are developing a natural antibiotics, you want its potency and selectivity to be comparable to that of synthetic ones, so that there would not be a need to use a synthetic antibiotic. And this is what they found with one of the nanoparticles using Lantana camara, this plant species. Right? So that is basically my presentation. You know, it's beyond the scope of this presentation to examine all the plant. Because of time, it's impossible. But the important thing, the take-home message is that there is a need to intensify green chemistry nanoparticles synthesis and to use them in nanomedicine, because you have an abundance of plant material. It's bio-friendly, it's environmentally friendly, and it's cheaper. You can scale it up and so forth. So there is need for further research to, you know, intensify. There's a need to intensify research for based on plant-based nanoparticles, and I'm sure that scientists will find some very useful results. They will make some very useful discovery. And with COVID around, it's worthwhile to use some plant-based nanoparticles and see whether they have an effect on the SARS coronavirus, whether they have an effect on that. They should. So it should be one of the molecule plant-based nanoparticles used to investigate, or as a treatment. They should have, research should investigate the use of plant-based nanoparticles for a COVID-19 drug. Thank you all.